All right, well, we're ready to begin Chapter 13 because we now all know what we're going to pretend we all know what replication, transcription, and translation are. Okay? In this chapter, we're going to focus on viruses, but we're also going to talk a little bit about viroids and prions while we're in this chapter. Viroids and prions are not viruses. They really don't look anything like them or act in any way similar to them, but we kind of lump them all together in one chapter because all three of these are considered non-living infectious particles. Okay? Viruses are actually the most complex of the three, and you may think complex means impressive. It's actually more impressive that some of the simpler ones, like prions, can do what they do to you. So, and we're going to go through all that as we go through the chapter. But let's start with viruses. These are your 6 or 7 characteristics of viruses that you all just kind of need to know. You need to be able to spout them off. Number 1, they are too small to be seen with a light microscope. Okay? And the way that was first described, it was described as viruses are filterable. Because this goes back to your 1800s, your 1700s, when we were first discovering there were really tiny cells, which we now know are bacteria, that were making people sick. Well, when we first started finding things that were making people sick and we were trying to isolate them, we would filter them. Okay? And if it was a bacteria that was making people sick, the filter would catch the bacteria. But then all of a sudden we were coming in contact with these diseases that we would try to catch the bacteria that were making them sick, and we couldn't catch anything. Well, it turns out these were viral infections, and the viruses were just going through the filters that we had. So that's all that filterable means, and they love to put that on exams. If you take multiple choice exams, they're going to throw that in as a choice somewhere, all those standardized tests. And that just simply means they're so much smaller than bacteria that if you use a filter, the filter is going to catch the bacteria, but most of the time the virus is just going to go straight on through just because of its size. Okay? What makes a virus considered not alive is that outside of the human body or outside of a living cell, they can't grow, they can't reproduce. That's what we mean by they can't be cultured. So we use agar, those petri dishes in lab all the time to grow bacteria, right? All we got to do is touch a piece of bacteria on and that bacteria starts growing like crazy. You could rub virus all over that and nothing's going to happen. That virus is just going to sit there indefinitely. If the virus has a cell to go into, then that virus can reproduce unbelievably fast. It can't do anything outside of a living cell. Okay? They are considered obligate intracellular parasites. And that's kind of related to this one that we were just talking about. Obligate means what? Absolutely has to be an intracellular parasite. So the only way it can exist is feeding off of a cell, living inside of the cell. When it's not inside of a cell, all it is doing is just floating through space until it finds another cell that it can infect. Okay? When we talk about bacteria, I spend a lot of time harping on all the good bacteria, right? There aren't really any good viruses. There are some that aren't human viruses that we have learned how to use to our advantage, but they're bad to something, whatever they infect. There's no such thing as, oh, I've got a good virus living in my body. That doesn't exist. Okay? Viruses contain either DNA or RNA. That is one of the weirdest characteristics that we've ever discovered about a virus. Can humans live with just DNA or just RNA? We have to have both of them. Every living thing on Earth has DNA and RNA inside of it. Viruses do not have to have both. Okay, they can be just one or the other. They're never both. All right, all viruses contain a protein coat surrounding the nucleic acid, and we're going to look at this in detail in a minute. Sometimes they have an envelope on the outside, but more of that in just a minute. As we've been discussing, they use the host cell machinery to replicate, meaning that they go into our cell, and they hijack all of our materials to make more of themselves. The last characteristic is one that some of you would probably argue with. viruses have a very narrow host range. That means a virus cannot naturally jump from species to species. Okay? If it's a human virus, your dog can't get it. If your dog has a virus, you're not going to get it. And there's always somebody who say, yes, my dog had the runs, and then I got it. That was most likely a coincidence. All right. There are cases where viruses have jumped species, but when that happens, the virus had to mutate to be able to do that. It can't just naturally do that. Right. And so when they do that, is that a big deal? It's a very big deal, right? Swine flu, that was a big deal a few years ago. Not that the swine flu is any worse than the normal flu. It's just different. None of our stuff could work with it because that's not a natural human virus. Okay, it's normally a pig virus. Bird flu is a bird virus. It's not supposed to be able to infect us. Well, that's how we believe HIV started. We believe there's a real closely related simian immunodeficiency virus. We believe that it was a virus that only infected monkeys and that it mutated and somehow switched to a human host. Most people kind of giggle. That doesn't mean a human had sex with a monkey. No, most of your scientific community believes it happened in a lab. It happened in research labs. It's very common for people that work with monkeys for test subjects. They get scratched and bit a lot. Monkeys are not the sweet little things that you see, like when you watch, like Friends was a big TV show when I was a kid, right? And he had the pet monkey. Right? Pet monkeys are horrific pets that they bite and scratch and claw and all kind of stuff. And they think that's probably the most likely setting where it switched into a human virus, is during that interaction. Correct. You could mix your blood with a monkey's blood that had simian immunodeficiency virus, and you wouldn't automatically have HIV. They are different enough. Right. But that is where we believe it came from. And that's honestly where a lot of our really nasty viruses came from mutations from other species. Yellow fever, that was a mutation from a monkey virus. Okay. Next to monkeys, we're most similar to cows and pigs. So if there's going to be a virus we're going to get that's not originating from a monkey, it's probably going to be a cow or a pig virus. That's a prion. So we're going to talk about that too as we get on into prions. Okay? All right, so those are just basic characteristics. Let's talk about something a little more interesting. First off, when I say viruses are small, let's get an idea of how small that really is. Okay? So on this picture, this big rod-shaped bacteria at the bottom, that's an E. coli. You guys have all seen E. coli in lab. It's one of the common ones that I give you. When we did our gram stains, do y'all remember those teeny tiny little red rods you saw under the microscope? Remember those? That was E. coli. So you think about how small that was. At our strongest magnification we have on a light microscope. If the E. coli was this big, these are the sizes of some of your viruses. And you can see the viruses, they look very different, right? They're not all shaped the same way. This candy cane is Ebola. You guys have all seen this candy cane because any time they put a virus in a movie, they show you Ebola just because it's cooler looking than a circle. But Ebola is the only one that has that candy cane, little hook shape to it. Tobacco mosaic virus, this was the very first virus ever discovered. Is that a human virus? What's that infect? Tobacco plants. Okay? Do any of you smoke? Yes, you all have tobacco mosaic virus in you right now. But it's not hurting you because you're not a tobacco plant. Okay? Every tobacco plant in the United States has that virus on it. If you want to do a really cool little experiment, you buy you 2 tomato plants. They're really closely related to tobacco plants. Close enough that you can infect the tomato plant with tobacco mosaic virus. So take you a cigarette. If you smoke, inhale a little bit and then blow the cigarette smoke on one tomato plant and not on the other. And you'll see the one you blew the smoke on will wilt over and die. And the other one will live because you gave it tobacco mosaic virus. If you don't smoke, you can take a cigarette and crumble it up and put it on one and not the other, and the one you put it on will die. A lot of people that smoke have a hard time getting their garden to grow And that's because they have that virus on them and in them, and it's not hurting them. Mm-hmm. I've never heard of it. It would have to be a hellacious shock for it to work. Like it's going to kill your cells just as fast as it could kill the other cells. A lot of that kind of stuff and a lot of your alternative medicine, and I'm not saying it's all bad, and that's not what I mean by it, but a lot of that stuff, it works by making you think it works. And a lot of your illness a lot of times comes from you think you're sick, right? Have you ever thought you were going to get sick and you got sick? Yeah. Your brain can do that to you. And I think that's where a lot of those treatments work, is you think you get better. But, I mean, I don't know for sure. I don't really know exactly what you're talking about. But I always thought that was kind of neat that people carry around tobacco mosaic virus. Okay. We're not talking about prions yet, but we're going to. While we got this picture, I'm going to show you the size. Prions are really, really simplistic, but you see they're not the smallest. The smallest infectious particle on Earth would be a virus. Bacteriophages, we're going to talk about those a lot. Anytime you see the word phage, you automatically think, okay, that is equivalent to virus. A phage is a virus. Okay. What do you think a bacteriophage infects? Bacteria. There are many, many viruses that infect bacteria. Okay? And those don't usually hurt us, of course, because they're infecting bacteria, and we don't really care if they infect the bacteria. We use bacteriophages to our advantage. So we'll take a bacteriophage and we'll put human DNA in it and then infect a bacteria in the lab with that bacteriophage. It puts the human DNA in the bacteria. Then the bacteria will replicate the human DNA for us and we can purify it back out. It's just really, really neat. We do a lot of our scientific work and things we know about viruses we've learned from the bacteriophage because they don't hurt us. Just point out a few other interesting viruses while I got the picture up here. This is the rabies virus. We say it looks like a bullet. That's how it's characterized. They actually will run tests, look at it under the microscope to see if it has the bullet shape to it to tell you have rabies. What does a rhinovirus cause? Common cold. Common cold. Okay? Real, real common. We have all had one of those at some point in our life. So I just wanted to kind of show you some of the sizes. All right? So now let's look structure. In order to describe a virus, we need to learn a word, and that word is a virion. A virion is a complete, fully developed infectious particle. So when I tell somebody that, yeah, I have a virus, that's not really proper terminology. You actually have virions in your body if they're making you sick. Okay, but that's really kind of minute detail as to how you describe it. As long as you can say virus, I'm pretty satisfied. But let me kind of just draw a typical virus for you. Come on, computer. Cooperate with me. Okay? Every virus has two parts. Okay? It's got the nucleic acid in the middle. That's what we call the core. What are the two types of nucleic acid? Deoxyribo or ribo, right? DNA or RNA. They can be one or the other. Something really neat about a virus, their DNA can be single-stranded DNA or double-stranded DNA. How's our DNA always found? Double-stranded, right? They don't always have double-stranded. They can have single-stranded or double-stranded. They can have RNA. Their RNA can be a single strand of RNA or a double strand of RNA. Our RNA is always single-stranded. Okay, so that's one difference between us. Around the nucleic acid core, they have what we call a capsid. The capsid is made of protein. Okay? And I'll show you a real picture in just a second. Okay, so far we have DNA or RNA, protein wrapped around it. Then that's the two components you always have. Sometimes you have another coating on the outside. That's what we call the envelope, and it's made of lipids. Very, very basic molecule. Always has DNA or RNA with some protein around it. The protein is not fancy. It is just a polymer of the same protein over and over and over again. You have billions of proteins that make up your body. He's got one type. Okay? Then just every now and then they have that extra fatty envelope around the outside of them. Okay? So let's look at some of the real pictures. Okay, I really need that to go away. Okay. What on earth? Yeah, see, the computer hates me, right? It's not just me. All right, well, it's just going to be there, I guess. Okay. It really drives me crazy. All right, so here's some real pictures. A little bit better drawing than mine, not much. So this purple stuff on the inside, it's your nucleic acid core. Little blue ball structures on the outside, that's making up your capsid. This is an example of a polyhedral capsid. This means the capsid has 20 flat sides. It looks like a diamond. Okay? And if I flip back and we look back at this original picture, I know they're small, but do you see how they kind of have that diamond shape to them? Those are all polyhedral. It's a real common shape for the capsid. Each one of these little balls is supposed to represent one protein. And it's kind of complementary in the way that it's developed. The virus will make one little capsomere, one little protein, then it makes another one and hooks it together. Then another one and hooks it together. And it just keeps sticking all those little structures together until it makes a nice perfect coating around that nucleic acid. The only other shape option we have for a capsid is this one. This is called helical. It's written under there. Helical just means that instead of the diamond shape, the capsid has almost a barrel shape to it. Still the same thing. You're going to have the nucleic acid in the middle. Okay? Both of these are what we call naked viruses. Naked meaning they don't have an envelope on the outside of them. It doesn't mean there's something wrong with them or they're any less virulent or anything like that. They just happen to not have an envelope. If the virus wants to have an envelope, it takes its nucleic acid core with the capsid wrapped around it, and it puts all of that inside of a little fat bubble. Sometimes the fat bubble has little spikes sticking out. Again, that kind of just depends on the virus. But I want you to understand the three basic structures. Does that make sense? Polyhedral and helical are both naked, or they can be enveloped. Okay? So how many of you would have thought that this crazy-looking structure right here is what a virus looks like? Right? That you think that's what viruses look like because that's the picture we show you in every class where we just kind of pop it up there and say, this is a virus, and now let's go talk about something else. The only reason we show you this one is because it looks cool. It's just a funner picture to show somebody. There's no such thing as a virus that can make a human sick that looks like this. The only viruses that look like that are the T7 or some of your T-even bacteriophages. That's a virus that infects a bacteria. Okay? But if you just, that's driving me crazy. Let me see if I can get that thing to go away. It still won't go away. Okay, I'm sorry. So underneath this drawing that will not go away, what does this look like up here? It looks like this, right? Does everybody see that? Okay. This is your nucleic acid core. It's always DNA in a bacteriophage. This blue part of it, that is the polyhedral capsid around it. All this junk down here is just some extra stuff that helps the bacteriophage infect the bacteria. And the way it does it, it's pretty neat. He uses his little tail fibers as legs, and he attaches to the bacteria with his legs. And once he attaches to the cell wall of the bacteria, he kind of squats down. And when he squats down, the base right here has little sharp pins. Those pins will pierce into the peptidoglycan cell wall of a bacteria, and then the DNA will move through the little sheath and squirt into the bacteria. And then all of this junk, that's just made of proteins. It just falls off. Now the virus's DNA is inside of the bacteria, and he can do what he wants to do. He can replicate, and he can make more of himself. Hmm? What? Yeah, they don't have to have envelopes. Just some of them do, some of them don't. Like some of us have long hair, some of us have short hair. So, I mean, kind of same. It's not necessary. This is just what we call complex structure. Every now and then you have a random virus that will develop a complex structure, and it will be specialized for whatever that virus can infect. Only reason I even point out how this guy works to you is because when we start talking about how they replicate, we're going to start with him because he's the simplest one. He infects bacteria. Of course, a virus that infects you has to be more complicated, right? Because you're a lot more complex than a bacteria is. Okay? All right, now as we keep going, you guys have got to stop me if at any point this does not make sense so we can discuss it. That's just driving me crazy. I've got to try to get rid of that. Okay, yay, it went away. All right, so this is what we went over last week when we did our biology review, right? And you all understand this. What we're ready to talk about now is how the virus can get into your body and make millions and millions of copies of himself. Okay? So you have to understand these things. So everybody good? All right. Humans can do replication, DNA to DNA. Humans can convert DNA to RNA. And humans can turn RNA into proteins. That's the basics. Humans cannot make a copy of RNA from RNA or go in reverse and do RNA to DNA. So anytime you see the top three things going on, you have to automatically understand that's something a human can do, so the virus is using what the human has. Anytime you see RNA going to RNA or RNA going to DNA, that's something humans can't do, so the virus must be using his own stuff to do that. That's clear, right? Okay, because some of you aren't going to like this once we get started. All right, so we're going to start with the simplest way a virus can infect a cell and replicate to make copies of himself. Okay, and that's called the lytic cycle. We're going to use the T-even bacteriophage. T-even meaning it can be a T2, a T4, a T6. That's just what even means. We're going to use the bacteriophage to kind of demonstrate how this happens. Then we're going to slowly move into viruses, animal viruses. Okay, so lytic cycle. What does lytic mean? What does lyse mean? Cell breaks, right? So you should automatically assume, okay, that means at the end of this, the bacteria that's been infected is probably going to lyse. He's going to die. Okay? It's considered a cycle. What does cycle mean? What you start with is what you end with, right? Anytime we go through these, you have to keep in mind the virus is going to have to look at the end exactly like it looked at the beginning. Okay? So if I have a virus that goes in with a polyhedral capsid and a single strand of DNA, I'm making viruses with a polyhedral capsid and single-stranded DNA. Did that sentence make sense to everybody? Whatever it looked like going into the cells, what we're going to make, and that's what they're going to look like when they come out. So we've got to go through steps to get to that. Okay? We're going to go through 5 basic steps: attachment, penetration, biosynthesis, maturation, and release. Okay? Sometimes you will hear or see maturation also called assembly. It means the exact same thing. And your textbook even flip-flops back and forth. It's the same step, okay? All right, so I'm going to flip to a picture. You guys have words to study with, but I want to flip to this picture to kind of show you what's happening, okay? So here's the lytic cycle on just this side we're looking at right now. This brown thing, that is our bacillus-shaped bacteria. This crazy-looking thing right here, that's our phage, our bacteriophage, our virus. Okay? Step 1 is attachment, and that means exactly what it sounds like. The virus has to bind to the bacteria. That's obvious, right? If it's going to get into the cell, it has to have a way to bind. And that's where that host specificity that we were talking about at the beginning comes into play. This bacteriophage can float by every cell in my body forever, and it can never infect me because it cannot bind to one of my cells. Those little tail fibers, those things that look like legs, they specifically bind to peptidoglycan. So they can't do anything until they find peptidoglycan to bind to. So even though attachment seems simple, it's a really important step. It's a very specific step. It has to be able to grab a hold. Now that the virus has attached to the cell, step 2 is penetration. Penetration means the nucleic acid of the virus has to penetrate or go into the cell that is trying to infect. Okay. With the bacteriophage, this purple thing right here, that is the DNA of the bacteriophage, and it alone goes into the cell. Now it has penetrated the cell. We're right here. This is your bacteria. The purple, that's the DNA that the bacteria had, because it's a living cell. It had its own DNA. The pink, that is your viral DNA. At this point, we've done attachment, penetration. Now we have biosynthesis. The cell is now going to make everything the virus needs. Okay? So what does that virus need to make? When that virus attached right here, he had a piece of DNA, right? So if he's going to make more of himself, what's he going to have to do to this piece of DNA? Make more. He's going to copy that DNA. Okay? What's wrapped around the DNA was proteins. So he has to make more proteins. Okay? And that's what it's trying to show you right here. Now we've made more and more DNA, and we've made more proteins. How did that virus make copies of DNA inside of that bacteria? He used the bacteria's DNA polymerase, right? He used the bacteria's nucleotides, and he just used everything that was in that bacteria to make copies of his own viral DNA. How did he make his proteins? Once he made his DNA, he converted them into messenger RNA, right? Then the messenger RNA fed into the ribosome, made proteins. So now do you see why we went back and reviewed all of that? All that happens in this one little step right here. Okay? So what do you think maturation, or what we call assembly, would be? Putting it all together, right? Real basic, makes sense, common sense. I copied a lot of DNA, it's right here, and I made some proteins over here, but I've got to make sure it's put together correctly. The DNA has to be put inside of the capsid, and I got to attach all those legs and everything to it because the bacteriophage has that crazy shape to it. Okay? Last step is release. Since this is the lytic cycle, they literally punch a hole in the bacterial cell wall, and they come flooding out of the cell. What happened to my bacteria? He died in the process. Okay? So I have a little animation. Let me turn it off. It's really short. But I think these help for you to be able to kind of see what's going on. Okay? So what's this you guys can see right here? That's the capsid. It's a polyhedral capsid. So inside of it's going to be what? The nucleic acid, DNA or RNA. Okay? So viral genome. What does the word genome mean? The genetic information, the sequence of your genetic information. So that's your viral DNA or RNA. And if you guys like these videos and you think they're helpful, I'll upload them on Blackboard for you to watch them. Okay? You'll just have to let me know. So this is representing our host cell. Okay? So what's the first thing that we have to do? Attach, penetrate, right? So now we did attachment, penetration. Those were the easy parts. That's why they go through them so quick. Now we have biosynthesis. So if this is a piece of DNA, now this is assuming a eukaryotic cell. So the DNA goes into the nucleus. You make lots of copies of the DNA. I'm going to let y'all watch, and then we'll go back and talk about what happened. All right, so let's slow it back down a little bit. So here's the viral DNA. Why did it go into the nucleus? Because that's where we copy our DNA, right? So the viral DNA goes into the nucleus, and now it's going to use the host cell's DNA polymerase and make copies of itself. So here you go. It's going to make lots of copies of itself. Now the DNA is turned into a different molecule, and that molecule goes out into the cytoplasm. So what's that red stuff right there? It's the messenger RNA, right? So after the viral DNA was copied inside of the nucleus, we made copies of all of that DNA and turned it into RNA. Why did the RNA go out here in the cytoplasm? To make proteins, because that's where the ribosomes are, right? Okay. So now we make proteins from the RNA. Why do the proteins go back into the nucleus? They got to get their DNA, right? Can DNA ever leave the nucleus? No. So if those proteins are going to go down in there, we're doing maturation. We're assembling the viral particles. Those proteins have to go in there and get the DNA so they can then bring it out. So they go in, we mature, we wrap everything up. Why did we not take the viral RNA with us? What did this look like when it went in? The little polyhedral capsid with DNA, right? He's not going to take that RNA with him. The only reason he made the RNA was to be able to make his proteins, right? So he doesn't need to take that with him. He was just using it to make his proteins. Now those viral particles are done. Here they go. Release. Cell dies, all the viruses come out. Does that make it better? See it a little three-dimensionally? Usually, to me, it does. Well, it could be either. The drawing I was showing you was a bacteria. This was a eukaryotic cell because it had a nucleus. But it's still the same effect. I just wanted you to be able to understand the biosynthesis. I can't draw it like I can show it to you in animation. Okay? So we're good. Lytic cycle makes sense, right? All 5 steps make perfect sense. Okay? Do you think all viruses would kill their host cell? What would be the downside of killing your host cell if you were a virus? You're going to run out of host cells to infect, right? If I'm a virus, the only way I can survive or live is to get into a host cell, right? If I kill them every time, well, then I'm going to eventually run out of host cells, and then I can't do anything. So there is an alternative to the lytic cycle. There is something called the Lysogenic Cycle. With the Lysogenic Cycle, the host cell does not die immediately. The virus takes his DNA and sticks it into the host cell's DNA. Okay? So step 1 and 2 are the same. Attachment works the same way. Penetration, the viral DNA coming in, works exactly the same way. For it to be lysogenic, this is your step that's different. The viral DNA becomes a prophage. This is the viral DNA. This is the bacterial DNA. It's now one big piece. Okay? This bacteria can still live perfectly fine with that viral DNA incorporated into his DNA. What do bacteria love to do? Replicate and divide, right? So now this bacteria with this little piece of viral DNA in it is going to keep growing and dividing just as fast exponentially, just like we talked about, right? Every time he replicates his DNA, he's also going to replicate the viral DNA, right? So this virus is just hanging out dormant, hiding in the host cell DNA, being copied every time that host cell divides. Days later, weeks later, months later, years later even, we now have this huge collection of host cells, all with viral DNA in it. The virus can come, the DNA can come back out of the host cell DNA, switch into a lytic cycle. This is exactly how HIV works. Okay? So let's say now viral DNA comes out. Instead of just one cell going through this part of it, making lots of copies of the virus, eventually the cell dying as the viruses release. This isn't just one cell now, right? This is millions of cells. And so instead of having 10,000 viruses, you have 10,000 times millions all coming out at once. Okay? That's exactly how HIV works. This is why there's no treatment, or especially no cure for HIV. Okay? We can't kill the HIV virus because it's sitting right here inside of the human's DNA. So the only way to kill it would be to destroy the human's DNA. Not always, but sort of. You're on the right track. So think about it this way. Which one of these is going to make you feel sick faster? Which one's going to make you feel sick? The lytic's going to make you feel bad. So when you get something like the flu, that's a lytic cycle. You get the flu, you don't really feel it for a day or two, then you feel like death for a couple days, and then you're fine, right? You've went through a lytic cycle. Once you become fine, that's when your immune system has killed all the viruses. When you feel really bad, that's when the viruses are killing your cells and multiplying. Okay? Somebody can get HIV and have absolutely no idea they have it for 10, 15 years. Okay? It just depends on the person. It depends on the strain of HIV that they got. Some strains are more virulent than others. Some of them burst back out and start doing the lytic cycle faster, and that's the people that start feeling the symptoms switching into AIDS sooner than others. The medications we take for treatment of AIDS, they're not killing virus at all. They're coming into this area and stopping it from switching back into the lytic cycle. They're just keeping it under control, keeping it from replicating anymore. So when people take treatment, take drugs for HIV, they still have HIV. They're still very, very contagious. The scary part of all that is, can you look at them and tell they have it? There isn't a single sign in the world that you can look at and tell. At least gonorrhea or chlamydia or herpes, you can, for the most part, at least you kind of see that. HIV, you can't see at all, and it's deadly. And the other's just inconvenient. Okay. All right, so I think I have one of my little, I think this animation shows you kind of the switch back and forth between lytic and lysogenic. Let me show you this one. I haven't put them on there yet, but if y'all like these, I'll put them on there where you can watch them. And there's little questions you can answer if you want to, but you don't have to. I just like the videos. Let me make sure this is what I think it is. Yes. Okay. Sorry, he's very wordy. Let me just turn him off. All right, so lytic cycle again. I know we just watched it, but this time the viral DNA is green. The host cell DNA is purple. Okay, attachment, penetration. This time they're also showing you this DNA is double-stranded. The DNA they showed you before was single-stranded. It can be either way. That just depends on the virus. Okay? So attachment, penetration. What's next? Biosynthesis. Good. We got to copy the DNA, right? So we're going to see lots of pieces of green. Eventually. Told y'all he was wordy. I think I'm wordy. Okay, I really hate that part of the animation. The host cell DNA doesn't disappear. They just move it out of the picture so you can see what's going on. Okay, but don't get that in your head that the host cell DNA magically disappears. Okay, biosynthesis. He made lots of copies of his DNA. Now what's he going to do? Make the RNA, make the proteins, assemble everything. It's lytic, so we're going to break open the membrane and release. Yeah, because then now they're showing you a bacteria again. And you just have to kind of keep up. That's a really important distinction, and we're going to get to animal viruses in a minute. You'll be able to clearly tell the difference. Okay, so here's the bacteria. attached and penetrated already. This is our viral DNA. It incorporates into the host cell DNA. Now this is called a prophage, meaning the viral DNA is just inserted in there. Now that cell is going to just start reproducing. Every time the host cell reproduces, he's going to automatically make a copy of the viral DNA, the prophage. And one thing that's really neat, they don't all necessarily come out of lysogenic into lytic at the same time. Some of them will stay lysogenic, some cells will switch lytic. So you start killing a few of these cells, at the same time you're still hidden in some of the other cells. And so it's just, these are the type of viruses that there is no cure for. Your immune system can't do anything about it. I think we froze up. We're not moving anymore. There we go. So now our host cell starts replicating. You can see every time the host cell replicates, it's a new cell that has that viral DNA in it. And that can go on, as I said, for years. No one's asked me yet what makes it switch back to lytic. Now, somebody always asks me that question. I can't answer that. No one can answer that. We don't really know because it's really hard to understand how could the virus know and decide. How can just a piece of DNA decide when it wants to do something? We don't know. All we know is eventually they do switch back to a lytic cycle and start killing their cells. Okay? So how are we feeling so far? You're halfway through. You have the basic information at this point. You know the 5 basic steps. You have an understanding of what occurs at each of the 5 steps, right? Animal viruses are going to be a little bit more complex. Okay, so we got to make sure we understand. Okay. So now I put all of this stuff in words for you in your notes so you could study it. And so you don't have to sit here and frantically write stuff down. But what I'm going to do is I'm going to narrate an animation. And then we'll let you kind of go back and look at the words and make sure everything makes sense to you. So now as we watch, this is showing you infection of viruses into animal cells. So this is how it infects your body. So you see the animation is going to be a little bit more complicated. Attachment and penetration is not as simple. There's 3 different ways an animal virus can attach and penetrate into your cell. That depends on whether or not the virus is naked or enveloped. And then there's 2 options, membrane fusion and phagocytosis. So let's look at those first. Okay. Everybody clearly understands what we're watching, right? This thingy up here, that's your virus. This one's naked. It's just got a capsid with your viral nucleic acid in it. This is the membrane of your mammalian cell. Now this is an animal cell, so there's going to be a nucleus and all that involved, right? Okay? So let's look at our 3 options for attachment and penetration. Turn him off. Okay? So here comes our animal virus. It's not normally this slow. My internet connection, for some reason, is not really happening today on this computer. Here we go. So if it's a naked virus, meaning not enveloped, it can just bind, attach to the membrane, and spit the nucleic acid in. That's exactly what we've been seeing, right? That's the basic way. But if the animal virus is enveloped, it can't just do that. Because if it's enveloped, it's got a fatty membrane around it, right? So we're going to have to do something with the fatty membrane in order to get the virus into our cell. It's jumping. I don't understand what the problem is. Okay, it's completely skipped one. Okay, so here's membrane fusion. The line around it now is the envelope. What is your membrane made of? Come on, what's your plasma membrane made of? Phospholipids, right? Your membrane's made of lipids. What's the viral envelope made of? Lipids. So membrane fusion, the virus's envelope, those lipids just fuse and enter into your membrane. All right? And now as we watch what happens, the whole capsid comes in. If this occurs, now we have to have an extra step called uncoating. We have to get that off. Here's the crazy part. Our cells do that for the virus. So once the virus binds and his little membrane fuses with our membrane, his capsid comes in. Our cell says, oh wait, we don't like to have DNA wrapped with protein inside of our cell. So our cell machinery goes over there and our lysosomes help break that down and release the viral DNA or RNA out so that it can now go in and infect our cell. So we help it. Okay? So that's just uncoating. That's just an extra step we have to have. Phagocytosis means what? Engulfing. Our cells will engulf a virus. They have ways of tricking us into doing that. So now we engulf the whole daggum thing. So now, of course, we have to uncoat that because we're not supposed to have all these fatty things and protein-coated things in our cell. So we uncoat that for the virus. And now we just have nucleic acid on the inside of our cell. I'm sorry it's so jumpy, guys. So does that make sense, the three ways that it can attach and penetrate? If it's naked, it just binds, spits the DNA or RNA in. If it's enveloped, the fatty envelope either has to fuse with our envelope or the whole thing comes in. Once we get it in there, our human cells uncoat it, release the nucleic acid so it can now infect our cells. Okay? It uncoats the lipid off and the protein off. It does it in two separate steps. Okay? So now we've done attachment, penetration, and uncoating if necessary. Now we're ready for biosynthesis. Okay? Look how many different options there are for animal viruses. Animal viruses can be ssDNA. What does that mean? Single-stranded DNA. They can be double-stranded DNA. All right, now there's a plus and a minus RNA. The plus RNA is what we call a sense strand of RNA. The negative RNA is what we call an anti-sense. So what does that mean? If something makes sense to you, what does that mean? It makes sense. It's logical. You understand it, right? A sense or a plus RNA, that's a piece of RNA that you can make proteins from. Okay? The negative one, your ribosome can't make a protein from it. Okay? How do you read a book? So I give you this piece of paper, how do you read it? From left to right. Okay, front to back. That's the way you read it. Would it make any sense if you read it the other way? No. Okay, that's all that means. Your ribosome can only read the RNA one way to make a protein. So if it's a positive, it makes sense to your ribosome. Your ribosome can read it. If it's negative, it's anti-sense. It doesn't make sense to your ribosome. So you're going to see the difference between them as we go through. Okay? Then there's double-stranded RNA. And then the last one is a retrovirus. Okay? So I'm going to play through this and explain it. It shouldn't make 100% perfect sense to you the first time. It should have to sink in. When we come to class next time, the first thing we're going to do is I'll take the entire board and I'll draw every one of them out and make sure you got it. Okay? So I just want you to see it all together one time today. And then we're going to go over it again and again. Okay. So we're going to start with our DNA ones. Why do you think we start with those? They're the easiest, right? Do human cells know how to deal with DNA? Yes, we do it all the time, right? So that's the easier one, so that's why we're going to start with that one. And let's back it up because things are retarded. Okay. So we've attached and penetrated. Here's the piece of single-stranded DNA. It goes into the nucleus, right? Because that's where our DNA belongs. Why do you think the first thing our cell does to a single strand of DNA is make a double strand? Because that's what we like, right? That's what we're used to. So the first thing we do, if it's single-stranded, always, we make it double-stranded. Okay? Now that that's a piece of double-stranded DNA, the enzyme DNA polymerase that's in our cell is going to copy that DNA. Okay? So it gets in, we make it double-stranded, and we make lots of copies of it, right? So now what do we need to make? RNA, so we can go make what? Proteins. Now the virus makes his proteins. The proteins go back in, grab the single strands of DNA, and now they can leave. That's the simple one, right? Everybody got that? So how do you think double-stranded DNA is going to be different? You're just going to skip one step. Everything else is exactly the same. Here's the double-stranded DNA virus. That double strand of DNA goes in. We just immediately start copying it. It was already double-stranded, so we didn't have to do the make it double-stranded step. Everything else is exactly the same. We copy the DNA for the virus. Then we make the viral mRNA. The viral mRNA goes to our ribosome. We help him make his viral proteins. His viral proteins go back into the nucleus. This time he picks up a piece of double-stranded DNA, and they're ready to go. Did you see the difference in what was picked up by the proteins? What went in, right? With the first one, it was a single strand that went in, so all the proteins did was get a single strand. Remember our rule? What it looked like going in, that's what it's got to look like when it comes out, right? That's one of those amazing questions that you would have to ask a virus. I have absolutely no idea how it knows. We do know there are slight differences in the backbone. So if you remember back with how your DNA is made, you've got sugar, phosphate. When it's viral DNA, there's extra little chemical molecules sticking off of that backbone, and that's the only thing we could think of as to how they know. But that's just like how we know. Our enzymes know which is the old part of the DNA and which is the new part, and that's just because there's little methane groups sticking off of the old part. It's just little extra chemical tells us which is which. Okay? There are probably times when the virus makes a mistake and picks up human DNA, but it can't do anything with it, so that particle is just going to float off and fall apart somewhere. Okay? So do we understand the DNA? Okay, let's do our RNA ones. So here is, why does it jump? Irritating. Okay, so this is what came in. The single strand of RNA, we've already attached and penetrated. So now we have a single strand of RNA in there. So what was the first step when we got the DNA in? Went in the nucleus, right? Is this going to go in the nucleus? No, it's a piece of RNA, right? He has no reason to go into the nucleus. Okay. Then what did we do with our DNA? We copied it, right? Do humans have an enzyme that can take RNA and make RNA? No, we do not, right? We take DNA to DNA and we take DNA to RNA. So this little red thing right here, that is a viral enzyme called RNA-dependent RNA polymerase. It used to be called Replicase, and I think they made it more complicated when they changed its name. Okay, so if it's an RNA virus, it has to bring an enzyme with it. The DNA virus, they didn't need to bring an enzyme with them. They just used ours. We don't have this enzyme, so that virus has to bring it with it. Also means he's going to have to make more of them. But enzymes are proteins. So if you have RNA, can you make a protein? Yes, right? So he can make all of this. Let's watch how he makes it. Okay. Since this is a single strand, what's the first thing we're going to do to it? It's what you think. Make it double-stranded, right? Okay. He's going to make his complementary strand using his enzyme that he brought with him. So now we have sense and antisense. Okay. Now he's just going to keep going, make lots of copies of both. Here he goes. That enzyme was inside of the capsid with the nucleic acid. Okay? So he made his RNA, he made his protein. So they do that like super fast. Everything else was so slow. Okay. So he uses sense RNA to make viral proteins, packaged up the plus strand and the enzymes and left. Here's the antisense, the negative single strand of RNA. He still had to bring his own enzyme with him because we do not have an enzyme that can copy RNA. Okay? He's going to use his enzyme to make the complementary strand. He's going to make the sense strand of RNA. Come on, little guy. There he goes. Makes some copies. Makes lots of copies of his RNA. Now he's going to make proteins. This time the protein's going to go get the green strand because that's what came in was the antisense and his enzyme, and he leaves. Well, just because they brought their own enzyme, that's not the only thing, right? They use our ribosome. They use our tRNA. They use our amino acids. They use our nucleotides. They use all of our stuff. They're just not using our enzyme. But they're still using all the other machinery. Okay? So this is double-stranded RNA. Still brings his enzyme with him. Okay? He's already double-stranded. He just starts making copies of his DNA, of his RNA. Once he gets a little collection of RNA built up, he's going to use his RNA to make proteins. Now this time he's going to wrap up both pieces because both pieces came in. And there they go. The last group, a retrovirus, is a piece of single-stranded RNA. What does retro mean? That doesn't mean old. A lot of people think it does. It means backwards. Okay? Retroviruses do the reverse of transcription. So what would that be? Taking a piece of RNA and turning it into DNA. Okay? Again, he has to bring his own enzyme with him because we can't do that, right? So he's a single piece of RNA. He brings his enzyme reverse transcriptase, and he makes a piece of DNA from his RNA. That piece of DNA is going to go where? Into our nucleus. Now that it's in our nucleus, we can make copies. We can make RNA. The RNA can go make proteins. Okay? So now here's the viral particles leaving with the piece of RNA, but what did he leave behind? His DNA. Guess what that DNA is going to do? He's going to become lysogenic. What does that mean? He's going to stick into our DNA. So this virus is pretty daggum smart, right? He made lots of copies of himself, and he left a nice little reminder that you're never going to get rid of. Hence HIV, herpes, things like that. Okay? All right, we're almost done. Now this should seem like a lot. I said that at the beginning, right? We're just going to look at all of them, and then we're going to break it down and go through all the steps again. There's two options for release once we're done, and that depends on whether the virus prefers to be a naked virus or an envelope virus. If the virus is naked, here he goes. Or there he goes. He breaks a hole in your cell, and off they go. It's a lytic cycle. It's going to kill your cell. Okay? The ones that are enveloped do it a little differently. They come up to the edge of your cell, and they've got to make an envelope, right? They need some fat wrapped around it. Guess where they steal the fat from? Your plasma membrane. So as they go, they just take a piece of your plasma membrane with them. Okay? And your cell's what? Your cell's perfectly fine. It is just a little factory making the viruses. Okay? So what do you think? Do you kind of have a new appreciation for how a virus can work in your cell?